Hello, everybody. Welcome. Good morning and good afternoon. And we appreciate you being here despite the time it may be in your location. I'm pleased to say that we have over 235 registrants from 36 countries registered for the second of 5 antimicrobial resistance and alternative agricultural research service. As the USDA's in-house non-regulatory research agency, the ARS conducts research to address agricultural issues of high national priority, and identifying solutions to AMR risks associated with agriculture is one of the USDA's and ARS's top priorities. As an agricultural research organization, ARS is uniquely positioned to identify AMR solutions to promote animal health, plant, environment, and public health while sustaining agriculture. ARS has a strong track record of solution-oriented, hypothesis-driven research that delivers innovative and sustainable solutions. ARS has a diverse, multidisciplinary workforce that conducts complex, long-term systems and longitudinal studies. ARS, AMR, and ATA research is an important part of 4 national programs: animal production and protection, crop production and protection, natural resources and sustainable agricultural systems, and nutrition, food safety, and quality. To build on existing research and formulate a cohesive, impactful approach to identify AMR and ATA solutions for agriculture, ARS hosted a Research Solutions for AMR workshop in 2021. Teams of scientists heard from key stakeholders and leaders in the field of AMR and ATA about the state of the science and to identify industry and regulatory research priorities on AMR and agriculture. ARS scientific experts across the One Health spectrum, animal, plant, environment, and food safety, collaborated to identify ARS priority research, needs, and priorities that will advance the science and identify solutions to address our 5 priority areas: risk, systems biology and detection strategies, mitigation, and communication, and that will advance our vision, which is to be the global leader for innovative, equitable, sustainable research solutions for AMR and agriculture, and our mission, which is to promote the resilience of agriculture to AMR for the health and safety of animals, plants, environment, and the public through cutting-edge research solutions and outreach. Over the course of 5 months, starting last month, ARS is presenting webinars highlighting ongoing research and having panel discussions focused on the topics. Today's session will focus on the priority topic of systems biology and detection strategies, and speakers will discuss AMR and agricultural systems, its detection and interventions from a One Health perspective, covering plants, animals, people, and the environment. Afterwards, there will be a Q&A session in which we'll be happy to take your questions, and we will have a panel discussion with speakers. We thank you for being here and look forward to the panel discussion after the talks. And as we begin, it is my great pleasure to introduce Dr. Jay Garland, our facilitator for today's webinar. Dr. Garland joined the EPA's Office of Research and Development in 2011. He received a PhD in environmental science from the University of Virginia and spent over 20 years working on NASA's effort to develop closed bioregenerative life support systems for extended human spaceflight. NASA recognized him for innovative technical achievements 4 separate times. He has worked on a range of topics, including methods for microbial community analysis, factors affecting survival of human-associated pathogens, and various biological approaches for recycling waste. Dr. Garland has completed visiting fellowships and professorships at the Institute for Environmental Science in Japan, the University of Innsbruck in Austria, and the University of Buenos Aires in Argentina. His current efforts focus on advancing innovative approaches to water infrastructure, including decentralized water reuse and mitigating risks associated with antimicrobial resistance in the water cycle. I've known Jay for over 20 years, and he is both my mentor and friend, and we appreciate him being here. And I'll turn it over to you, Jay.
Well, thanks, Kim. Appreciate the introduction. For those of you who don't know it, I hired Kim for her first job. So all of you in ARS, you have me to blame for the start of her career. So I'm really glad to be able to participate today in ARS's webinar series on antimicrobial resistance. I think this really is one of the major public health challenges of the 21st century, and I'm glad to be able to work with USDA on trying to develop approaches for dealing with it in agricultural production systems. We have a breadth of talks today, which speak both to the need for detection of AMR, but also the need to understand their role in the complex dynamics of food production systems. So I really look forward to today's speakers, and with that, I'll start by introducing our first speaker, Walt Mahaffey, from Corvallis, Oregon. He's a research plant pathologist with ARS in Corvallis. He has extensive experience transferring research into practical applications. That includes 2 biocontrol agents that are used in cotton and peanut, and also disease forecasting models. And what Walt's going to talk about today is his current research on looking at fungal resistance, both the detection and mitigation. So Walt, please, the camera's yours.
Well, thank you for the invitation to talk, and for everyone else for getting up, and those of you that are on the West Coast getting up at 6 a.m. and seeing this talk. Today I'm going to talk about working with grape powdery mildew and developing disease monitoring strategies and fungicide resistance. Now, one of the things that you're working in agriculture is that the fields are so large that we can't sample everything. So we actually need to start thinking about how the disease is not uniform or random. Where does it aggregate? And how do we target these areas in making decisions? Like here in grapes, we can see areas with high water table, nutrient accumulation in low areas, or even differences in the physical constraints of the agronomic system, where the tractor can't turn between these fields. So they have to actually go through, which causes a discontinuity in the crop phenology and the timing of the fungicide application. But we also know that plant diseases, the inoculum availability, it varies in time and space. The amount of the spore load changes as the season, but it can even change just from day to day and even during the time of day of when the spores are actually moving. And it also has a spatial component, and that because of topology and other constraints, that the epidemic is different from field to field. So how do we bring all of this knowledge into something that the growers can make a rational decision on when to time a fungicide and when it's best to use it? Well, we largely rely on scouting. This is a visual observation, and it's limited to a very short period in the pathogen's life cycle of when we can actually see disease. But the problem is that early in an epidemic, the odds are against that we'll even come across it. If you look here, there's a video of a simulation of a disease epidemic occurring, and these blue lines are transects. And then the dots are where we stop and look at the epidemic. This is very sped up. If you're seeing about a day a second in a lot of this. But the odds that we will actually even encounter, even with an extensive non-cost effective sampling strategy of 8 transects, is very, very low. The solid lines over here, they are where infection has taken place. So if we encounter the leaf, there's actually infection on it. But the dotted lines show what we can actually visually see when we encounter it. And you can see even out the 8 transects and, oops, there's a problem here. My scale is out of 5% disease incidence, we can't actually see disease in 50% of the encounters that we make. And also the problem is when we go out and look in the field, even highly trained scouts, they're just not very good at making that diagnostic visual identification early in the season. Now, as the epidemic develops, disease gets higher, they become better and better, so lower false positives at identifying the disease. And the question we started asking several years ago was, can we take a different component of the life cycle and actually use that to give us actionable information for the grower to time their fungicide applications or even their fungicide selection? So we started looking at pathogen dispersal. But one of the things that we observed, and this is in collaboration with Gary Grove at Washington State University, is that we have a temporal component in the spore load, and both the time of day that spores are available for detection and the time of year that they're detection. And again, I mentioned that there's differences from farm to farm. But the real problem with this, when we started looking at it, is the spore morphology is not diagnostic. And actually the equipment isn't overly cheap either. So we worked with Jennifer Fallacy here as a grad student that worked on this project. We started developing these molecular probes to go in and see if we couldn't detect the powdery mildew spores and identify exactly which one was there and present and how much. Initially, Jennifer's work showed us that we could detect ascospore release events, which is the movement from an overwintering inoculum to an effective inoculum, and then the conidial event later. So we could actually detect this, but the traps and the equipment to do this were cost ineffective for growers to start implementing it. So we started designing and building our own. And actually by version 4, we've gotten it down to where about $100 in parts. They can all be purchased online or 3D printed now. And it's only about 2 hours of labor to build. And while we were doing this, we developed a quantitative PCR that we could detect a single conidium 80% of the time from airborne samples. And we had an accurate spore concentration. We could accurately estimate spore concentration below 10 conidia that way. And one of the things that we found in developing this is that the type of master mix that you make is very, very critical, and that you have to really work with it and add far more nucleotides than what you would expect for most reactions in order to deal with the extreme background of non-target DNA that you're working with in these samples. Back in 2010, we started working on actually giving growers the power to do this assay in their kitchens, shops, whatever. We developed a lamp assay. And what we found were that the growers, when we gave them blind samples, they were actually really good at detecting it. But the problem came during the season is that they became overly anxious and they started imagining seeing the reaction. Even though we had at this time a fluorophore reaction, they were still overly cautious in reading it, and they also didn't really want to work with these samples anymore. They preferred to just have a lab process it. But over about an 8-year period of developing this, we worked with growers. We use this inoculum to time fungicides, so the detection, the first detection of the year, we would start the fungicide program, and then after that, based on the number of conidia, the growers would shorten or lengthen application intervals. And what we found over this 8-year period is we reduced fungicide use by about 4 applications on average. Now, there were some farms that we actually increased fungicide use on, but they were ones that were towards the extreme low end and managing the disease, or actually probably not managing the disease well. This cost savings ended up about $300 to $600 a hectare in savings, but more importantly, it reduced about 6 kilos of fungicide from being deployed into the environment. Currently, right now, with several commercial companies that have picked up this technology, I know of about 400 traps that are deployed throughout the West Coast. Now, we still know that the odds are against us, even with this trapping, because when we look at this simulation here, if you'll see, there's some pink dots that randomly deployed through here. We still know that even with the samplers, that we're not really above 50% probability of detection until we're at about 50 or 1% disease. Now in grapes, if we're reach 3% disease, we're in a state of crop rejection. And that's in the wine grape side. In the table grape, you can have crop rejection occur if any visible disease is available or is observed. So we started trying to do some working with some fluid mechanics, mechanical engineers working on fluid dynamics. And we started trying to understand how canopy geometry and topology affected plumes to understand what our samplers are actually looking at. And here's just a quick summary of about 7 years worth of research to demonstrate that with these samplers, when we place them in the canopy, the footprints are still quite low. We're only looking at about 25 meters from the location of the sampler. And that's in the upwind direction. In the downwind direction, we have very little area that we actually have a probability of sampling. But one of the things that we noticed is that there's all these workers always going through the field, and we started asking the question, can we use them as a sampler? So can we either attach something to them to collect spores, or ultimately what we decided is, can we actually use their gloves? So here's Sarah Lauder, a PhD student that's working on it, demonstrating what we came up with finally. Is that workers who wear gloves at the end of a row or in the end of a block that they're working in. They take this cotton swab, rub it all over the gloves, and we bring it back to the lab and process it. And what we found is that there's really not that big of a difference in the different types of materials that you can use to collect the spores. And they all seem to work quite well, even bare hands. And Sarah even found that with this particular pathogen, just soaking the hands or rinsing the hands really well in water works. Now, in some other pathogens that doesn't, but for powdery mildew, it seems to work quite well. So the question then was, does the glove detection better than the visual detection? Here is an ROC table looking at this. And what we found is that the glove detection has a lot more positives. We can detect the disease far earlier in the season at levels below 0.1%. And then we started to look at, well, here is a different example. This is LCA, because in this analysis, we don't actually know whether the visual detection or the glove detection is a glove standard. So with a latent class analysis, there isn't the assumption that either one is a glove or is a gold standard, like with the RCA analysis. And here what we found is that the glove swabs are actually far better. Now, the specificity in them is low for the glove swab, but this is an artifact of that, our visual detection. And the visual detection here, we actually confirmed every single observation through testing, doing a qPCR assay on the colonies that we found. So we're 100% sure that we have this accurately classified. But we found that the specificity is that the leaf swabs are actually, our visual ability is so poor that the specificity of the glove swab looks a bit off. When we look at this in terms of air samplers and compared to glove swabs, what we actually found is that they're pretty much doing the same job. They're giving us the same bit of information. So this led us to start looking at, can we use this data set that we've been collecting for just using spore detection to time fungicide applications? Can we use that same data set, the DNA, and start looking for the presence of fungicide resistance traits? And two of them that we're looking at is QOI resistance strobulorins, or FRAC 11, if you know your fungicide codes, or an azole resistance, DMI, or FRAC 3. But in both of these systems, we developed a hydrolysis probe or TACMAN probe system that we can quantify and detect the SNPs that are associated with this. And the QOI resistance, this is fairly straightforward in that there's a single SNP that we've shown is associated with resistance in the field. There's 100% agreement with the bioassays, and it's sensitive to a single spore. Now, with the azole resistance, it's a lot more complicated story because there's multiple mechanisms that are associated with it. There's a SNP, the Y136F, but there's also copy number that's associated with it. So we have a 4-probe system that works where we monitor a housekeeping gene so we can estimate the copy number of CYP51. And then we have 2 probes that allow us to determine the wild type versus the mutant type for the SNP of Y136F. This assay has about a 95% agreement with phenotyping. There's still one mechanism of potential resistance that we don't seem to be monitoring, and it's sensitive down to 20 spores. I'm going to give you the story of the QOI resistance just because it's simpler and more straightforward to talk, but all these samples we've also done with DMI resistance. In the last 4 years, we've sampled almost, or we've used growers to sample almost 5,000, give us 5,000 samples over the course of this time from California, Oregon, and Washington. And we have found in this, in many regions, they're approaching 80–90% of the population has resistance to QOIs. And then with DMI and looking at the assay, as I said, it's a little bit complicated. You have to know the frequency of the 136 or the Y136, the F136, the copy number of genomes. Then we use the delta as well as the CYP51 copy number. And based on these ratios, we can give you an idea. Here's an example in one vineyard, HO1, HO2, where we have an example of 2 isolates that were sensitive, but one only has the 136F, so it should be resistant. And the other one only had the 136Y, so it's wild type, but it has a low copy number of the CYP51. And here's examples of isolates that all have a high copy number of the CYP51, but there's one here where we don't have any of the Y136F, but it's still resistant. So this story is a little bit more complicated, as I was saying. But when we've looked at this in both the molecular characterization as well as the phenotypic characterization over the years, what we've found is that 62% of the population are resistant to both, but only about 2% aren't resistant to either. So fungicide resistance is a problem with grape powdery mildew, and it's growing. When we went back to all our spore samples and did historical analysis, we didn't have field failures until 2016 in this group of vineyards that we had been monitoring, but we could actually go back and detect the presence of QOI resistance starting back in 2013. So we knew it took a couple of years, or 3 years in this case, before it built to a problem where it caused field control failures. We also could distinguish within different vineyards how the QOI frequency was changing, which would show us that we could manage each vineyard differently using a fungicide combination. We know from looking over the sample period that every year the QI resistance starts as a low frequency. We actually have data that there's a fitness cost during overwintering, and it builds as the season goes. And part of that is because there's actually a fitness benefit to the A143A mutation during cool conditions. And then it's fairly neutral later in the season. So it can build, and particularly if you use QOI. And this is in the year, there's a little bit different patterns, and it deals with based on how QOI resistance are dealt with in the regions. And also with the sample type, and it kind of makes sense if you're using QOI or DMI resistance, that you are going to have a higher frequency of those populations that we can actually visibly see being positive. But more importantly, what this has allowed us to do is we can actually go in and monitor vineyards, and we can give them recommendations on when they can still integrate QOIs. And the reason that we're needing to do this is that there's very limited chemistries available for some other diseases that need to be managed. And we still need to keep QOIs in our toolbox in order to manage those other problems. So here's an example of where we timed a QOI application in a group of vineyards. And then, so we had an SDHI and a paired plot of QI, and we showed that we can keep disease levels at a quality that allowed for the quality standards to be met while still keeping the QOI resistance from becoming a real problem. Now, we've also examined, we've been looking the last year for the presence of Aspergillus fumigatus in all these samples, and we've actually yet to detect it. We know we can find it when we spike samples in, and we've looked at other pathogens that have similar spore traits, and we can detect those. So we know the samples, we should be able to detect Aspergillus fumigatus, but we just haven't found it yet in the system. This work, obviously, there's a huge team that goes behind it. A lot of collaborators, and particularly the funding sources. Hopefully I stopped on time. And yes, this was something they found in a vineyard. That's not CGI. See, there's one question in QA. Oh.
Yeah, that was, but Walt, that was about when the recording of the webinar will be available. So as we wait to see if there's any questions, I guess one I'll direct to you, Walt, is what's the response been from growers about the kind of the feasibility of this kind of fairly intensive sampling across time and space? How do you think it is? Is it cost effective to the growers?
It's cost effective because the glove swab, even though we're still kind of in the research mode, we've had some of the diagnostic labs that we've collaborated with to implement our inoculum monitoring work. They have actually picked up the glove swabs and the fungicide resistant assays, and they offer it as a commercial service now. And from my understanding is they get more samples than we do. And we're processing 1,500 to 2,000 samples a year for growers as part of our research effort.
Here's a question. I'll read it to you, Walt. It's the finding the growers are uncomfortable using the fluorophore assay formulated for their point of care testing and preferred laboratory test is interesting. I get the sense that the human factor has been very important with ARS working with growers. Was that completely unexpected, or can you share some insights on how you've worked with that human behavior aspect in your work?
No, the human behavior is something that we've incorporated. All of this research that you've seen is done with commercial growers. And actually, the concept of using the inoculum load to stretch application intervals was one of our cooperators. Over the years, we had always given him this information. He actually decided to start doing it. We'd kind of talked about it, but he just decided to do it and didn't tell me. which was probably a good thing because he grew $8,000 a ton Pinot, and he was using that to stretch intervals. And I think in the year that he told me, he hadn't made a single application that season. So the growers are intimately involved in the research, both in terms of refining the research ideas and in trying to solve what types of problems that they're interested. Like the lamp was an idea that they wanted to see, and then they decided, yeah, this is too much for us.
Well, thanks. I think we're almost at the end of time, Walt. I understand you won't be able to join us for a panel discussion, but appreciate your presentation this morning.
I'm at a conference moderating a session.
And you have a very legitimate excuse. So good luck with your conference, and enjoyed your presentation today.
Well, thank you, everyone.
Next, I was going to turn to the next speaker is Joanne Van Kessel. Joanne is a research animal scientist with the USDA-ARS in Beltsville. She received her PhD in animal science from Cornell University, and soon thereafter began working at ARS. And since that time, she's focused on the evaluation of Salmonella and Listeria in dairy cattle. And more recently, and what she's going to talk about today, is focused on understanding the factors that reduce both foodborne pathogens and AMR in dairy farms. So, Joanne?
Well, good morning, everyone. Thank you, Jay. It's a real pleasure to have this opportunity to share some of our team's research with you all today. And since we have such a diverse audience, I thought I'd start by sharing a few details about the U.S. dairy industry. Based on the 2021 milk production report by USDA, there are approximately 30,000 farms in the country and a little over 9 million milking cows. The farm numbers have actually been decreasing significantly over recent years. And if I'm correct, I think the number of cows actually has increased a little in the recent years. So each of these cows on average produces about 24,000 pounds of milk per year. And there's generally about a 30% turnover in the herd each year. And an often overlooked product of dairy production is the beef derived from these cull animals. This makes up to 20 to 25% of the US beef market. An important feature of the US dairy industry is the diversity with respect to farm size, climate, and geography. Herd sizes have been increasing, but we still have herds with fewer than 100 cows, and many farms have thousands of cows. These features all affect how the herds are managed, and the differences are really important to keep in mind as we look for strategies to reduce pathogens and antibiotic-resistant bacteria on dairy farms. Antibiotic use in food production animals, including dairy, is regulated by the FDA. It's challenging to quantify usage at the farm level, but Laurel Redding and colleagues from the University of Pennsylvania conducted a survey of Pennsylvania dairy herds a few years ago and published this in the Journal of Dairy Science. They showed that the most antibiotics are used to treat mastitis, and this was followed by treatment of subclinical mastitis via dry cow therapy. This lower chart is on an expanded scale to tease out the level of antibiotic use in calves for metritis in calves and for metritis in cows, and then also for respiratory disease and foot disease. The FDA also very strictly regulates antibiotic residues. In each tank of milk is tested before it gets unloaded at the processor, and any tank that's positive, the offending producer is charged for the full load. Likewise, culled animals are inspected and tested for residues at slaughter. And this approach is a very good deterrent, and the percent of infractions in both of these cases is very, very low. A few years back, when we started to consider the role of dairy production on antimicrobial resistance, we, in collaboration with colleagues at Penn State University, conducted a survey of 80 dairy herds in Pennsylvania. And we took composite manure samples from 4 groups on each of these herds, from pre-weaned calves, post-weaned calves, dry cows, and lactating cows. We cultured for Salmonella and E. coli and tested isolates for resistance to a panel of 14 antimicrobials. We isolated Salmonella from 13% of the pre-weaned calf samples, 25% of post-weaned samples, calf samples, 61% of the dry cow samples, and 64% of the lactating cow samples. These serotypes were represented in the isolates, and all of the isolates were pan-susceptible. This level of Salmonella was not surprising to us based on previous studies by our group, as well as many others. Although we didn't see resistance in these isolates, resistant Salmonella have frequently been isolated from dairy production systems. Just to give you an example, we participated in 3 of the NOMS dairy studies, which was run by APHIS, and where we isolated Salmonella from bulk tank milk and filters from dairy farms across the country. These are some of the serotypes that we isolated, and on the left were the serotypes of resistant isolates, and on the right were the serotypes of pan-susceptible isolates in all of these studies. As you can see, there's some crossover. There were some resistant Newports and pan-susceptible Newports, and the same is true for Kentucky. And one of the other comments I'd like to make about the Salmonella in dairy is just that some of these Salmonella infections will cause observable clinical disease in animals, and many others are almost a commensal infection and have little or no impact on the animals. If we go back to our farm survey, we isolated and tested at least 5 E. coli from each of the samples. And in contrast to Salmonella, we identified lots of resistance in these isolates. In this graph, the antibiotics are listed by their 3-letter abbreviations along the x-axis. And the colored bars represent the percentage of farms where resistance was identified to that antibiotic for each animal age group. The red bars are pre-weaned calves, the yellow bars are post-weaned calves, dry cows are the dark blue bars, and lactating cows are the light blue bars. For example, just to give you an example, ampicillin-resistant E. coli were isolated from the feces of pre-weaned calves on 57% of the sampled herds. As you can see, E. coli resistant to each of the antibiotics tested were isolated more often from calf samples than from the older cow samples. This graph helps us to describe the farm prevalence of isolates that were resistant to more than one antibiotic. The bars represent the cumulative prevalence of MDR E. coli on the farms. The color on the bar shows the prevalence for each age group for E. coli that are resistant to a certain number of classes. Going down each bar from the blue to the red indicates the number of samples that had E. coli that were resistant to progressively more classes of antibiotics, to 1 up to as many as 8 classes. As you can see, not only did the young calves have a higher prevalence of resistant bacteria, they also had bacteria that were resistant to more antibiotic classes. E. coli resistant to 6, 7, or 8 antimicrobials were only isolated from the calves and not from the older cows. Because E. coli represents a small proportion of the microbiome, we wanted to determine if our findings of resistance in E. coli are representative of the whole population. For this, we selected a subset of matched cow and calf samples from 17 of the farms, isolated the DNA, and sequenced the metagenome. The antibiotic resistance genes are identified by blasting the reads against the ResFinder database. The figure shows the percentage of total resistance reads for each resistance class. As you can see, the resistomes of most samples were dominated by tetracycline, followed by aminoglycoside, macrolide, and lincosamide streptogrammin resistance genes. And tetracycline resistance was by far the most abundant. You can see some differences between the calves and the cows, and as this is highlighted in these last 2 columns where the data are summarized for each group. We then looked at the relative abundances of total resistance genes in each sample and compared the calf samples to the cow samples. This figure on the left shows the range of these relative abundances for each group. These are the calves and these are the cows. And you can see across the resistomes, there's a higher abundance of resistant sequence in the calf samples versus the cow samples. And this just supports our results of the cultured E. coli work on these farms. The figure on the right shows the tetracycline, aminoglycoside, MLS, phenol, beta-lactam, and trimethoprim resistant sequences are all more abundant in calf feces, and there were no classes that were more abundant in the cow feces versus the calf feces. In summary, for this study, we concluded that the pre-weaned calf resistome is significantly different than that of the lactating cow, and that differences in resistome structures were driven by aminoglycoside and tetracycline abundance. We then turned our focus to try and identify the drivers of this high antibiotic resistance load in calves. We asked ourselves the question, why is resistance higher in calves than in cows? Studies over the years by different groups conducting work in different geographic locations at different times have shown that in dairy cows and beef cattle, younger animals shed a higher ratio of resistant E. coli to susceptible E. coli when compared with older animals. This has been shown using a single antimicrobial resistance gene, single antimicrobial resistance profile, and by us when we looked across an entire panel of resistance profiles. This has also been consistently demonstrated in swine and occasionally demonstrated in poultry and companion animals such as dogs, but the drivers of this phenomenon is not really known. One of the go-to explanations for the main driver of the presence of antimicrobial resistance in most systems, including animals, is the administration of antibiotics. This is partially true, and we know that the challenge of antibiotics can select for those organisms that are resistant to the antibiotic that's being delivered. However, we've seen in our studies, and others have repeatedly shown, that calves that are not treated with antimicrobials can still have higher resistant levels than cows. The basic observation is that calves are colonized with an E. coli population that's highly resistant, even when their exposure is feces from their dam or other cows in the herd, where the resistant E. coli population represents a very small proportion of the total E. coli population. So we asked the question, are there non-antimicrobial resistant features in the multi-drug resistant E. coli that facilitate the colonization in the calf gut? We decided to further analyze the E. coli isolates we collected during the farm survey. And to that end, we sequenced 264 isolates from each of the 4 animal groups. And from each group, we selected pan-susceptible, resistant, and multi-drug resistant isolates. This phylogenetic tree on the left shows the relatedness of our isolates, which are in red, and the black or gray isolates are reference genomes for each of the phylogenetic groups. Our isolates are spread across most of the reference groups. The 2 figures on the right show that the number of isolates assigned to each phylogenetic group. And as you can see, the most frequently detected group is B1, followed by A. Using the ResFinder database again to query our sequence data, we identified 951 resistance genes, making up a total of 45 unique genes. And this lower left figure shows that nearly 40% of the resistance genes were aminoglycosides. Tetracycline and sulfonamide resistance genes represented about 15% each. Beta-lactamases, phenol, trimethoprim, MLS, and fosfomycin resistance genes were also detected. And in addition, we identified about 50 chromosomal mutations known to confer resistance. We looked at the total gene content of all the isolates to see if there are any genes that are enriched in multi-drug resistant isolates that may explain the selective colonization of multi-drug resistant isolates in young calves. We compared the patterns of gene presence and absence among the MDR isolates from calves and cows to see if there's a difference in the gene content between these 2 groups. This plot is an ordination plot, so isolates that are more similar to each other are located closer to each other in the plot. When the isolates were grouped into MDR isolates from cows or MDR isolates from calves, the two groups of isolates were not significantly different from each other. So based on gene content, similar MDR isolates colonized both calf and cow gut. When we then compared the susceptible and MDR isolates and saw that there was a significant difference in gene content between the two, we thought that this may be due to the high abundance of resistance genes in the MDR isolates. So we removed these resistance genes and reran the isolates, and the results were approximately similar and showed that there is a significant difference in the gene content when the MDR genomes are compared to susceptible genomes. And these differences are not due to the presence or absence of the resistance genes. So based on gene content alone, the population of MDR isolates is different than that of the susceptible isolates. A Fisher's exact test was conducted on the pangenome to see which genes were present in the significantly higher number of MDR isolates, in a significantly higher number of MDR isolates, and those present in a significantly higher number of susceptible isolates. This is an approach to identify some, if any, of the genes that may be allowing the MDR E. coli to do so well in the calf gut. This column on the far left shows the genes that are enriched in either MDR or susceptible genomes. Orange bars indicate the genes enriched in MDR genomes, and blue indicates the genes enriched in susceptible genomes. The column is ordered with the most significantly enriched genes at the top to the least significantly enriched genes at the bottom. 75% of these genes are more abundant in the MDR genomes, and about 50% of those are plasmid-borne. In the second column are the resistance genes and it's not surprising that these are clearly highly enriched in the MDR genomes. They line up with the orange MDR genes on the column to the left. A very high number of these enriched genes were annotated simply as hypothetical proteins, which doesn't really give us much information about what they are or what they do. But one thing that really popped out was the number of siderophores, or iron scavenging and iron utilization genes, that were present in high abundance in the MDR genomes. Some of these were present in some of the susceptible genomes, but this was quite rare. We also conducted another survey of veal calves from commercial producers in Pennsylvania, and I don't have time to go into detail on this study, but we also isolated a lot of resistant and multi-drug resistant E. coli from the feces of these calves. And when we sequenced the genomes of both multi-drug resistant and susceptible isolates, we identified the same iron scavenging genes as significantly enriched in the MDR strains compared with the susceptible strains. It's possible that in the young calf gut, these iron utilization genes confer on the MDR isolates, which encode them, an advantage over susceptible isolates that lack these genes. We have ample evidence that multi-drug resistant E. coli are more abundant in young calves, particularly pre-weaned calves, compared to older cows. This phenomenon also translates to comparisons of the total resistomes of these animals. Young calves harbor a greater abundance of resistance genes than do older animals. We're trying to pick apart these phenomena from E. coli to the entire genome to find potential solutions to the problem of antimicrobial resistance and pathogen carriage in dairy animals. We see a strong association between multi-drug resistance and iron scavenging genes, indicating that available iron may play a role in the selection of MDR isolates in the calf gut. And this presents a potentially simple solution to this problem. Iron scavenging may be very beneficial to these bacteria, especially in the calf gut. Many other proteins enriched in the MDR E. coli could also play a role, but we chose to start with exploring the impact of iron because it makes biological sense. Calves are fed a milk diet which is relatively low in iron compared with the diet of older animals. So we hypothesized that the low iron input and concentration in the calf gut maybe at least one of the factors selecting for a higher ratio of resistant to susceptible bacteria in the gut of young calves. And as they are weaned off of milk, a decrease in this ratio is observed. We're currently doing some culture and animal experiments to test this hypothesis. Well, I said a lot of we during this talk, and there are a lot of folks who contributed to the work that I presented, and I particularly want to acknowledge Brad Haley, who's my co-PI and is the brains behind the genomics and metagenomic aspects of our research. And Sean-Woo Kim and Sergio Salahin are very critical to our team, our postdocs in our lab. And without Jackie Sagne and Laura Del Colo, I think a lot of our research would fall apart. And we enjoy a great collaboration with our colleagues, Ernest Hoving and Haling Springer at Penn State University, and they have contributed significantly also to the work presented here. So with that, I'd say thank you and entertain any questions that you might have.
Excellent talk, Joanne. There was one question on the from Q&A. Isn't it tetracycline a standard ingredient in commercial powdered milk used as calf feed? I think suggesting that might be one of the reasons why the prevalence for tetracycline resistance.
So tetracycline is really common across the spectrum when we look at different animal samples, and it's just a given that it's going to be there. And milk replacer is certainly a source of, is certainly antibiotic. Lots of milk replacer certainly does contain antibiotics, and the dairy industry has been moving gradually away from that. I think antibiotic supplemented milk replacer has been used less and less over the years, but without a doubt could be one of the sources. Yeah.
And I guess until there's any other questions, I was going to ask you about how receptive do you think the growers would be to interventions? I guess particularly when you brought up this relatively cost-effective potential intervention, right, of just changing the iron content of the diet, perhaps. But overall, what do you think that the interest or the willingness to accept interventions are from the producer side?
Well, I think you're right. I think one of our reasons for pursuing this iron supplementation mitigation is just that. It would be if we could ask producers to add small amounts of available iron to either the colostrum or the milk replacer that they're feeding, then I think that would be a low-cost, simple addition to their current management strategies. And I think beyond that, I think as with all other production systems, producers are working on tight margins, so any mitigations that are offered have to be something that are manageable financially and as well with the labor that they have on hand. So I think generally they're open to working with us, but there are a lot of caveats associated with that. And so we really need to have sort of good science, good data backed strategies so that it's much more convincing when we present our mitigation strategies to producers.
And are there any plans to implement this or do testing on the iron supplementation?
Yeah, we've conducted a short pilot study that we haven't analyzed yet, but we are certainly working on experiments, animal experiments, and some in vitro experiments with the E. coli isolates, looking at growth in different iron in medias with different iron supplementation or restrictions, and with the calves supplementing iron to their low iron diets, and looking at AMR bacteria loads in the feces.
One of the co-presenters, Danielle, had a question about, have you or your team tested the colostrum for AMR?
Tested colostrum for AMR. So we have not. I'm trying to think if I've seen any papers on that. There is, colostrum waste. Colostrum could contain resistant bacteria that if they're, mastitis, if the cows have mastitis with a resistant bacteria, there could be AMR in the colostrum, but I haven't seen any data on it in colostrum, certainly in waste milk. So waste milk is what farmers collect often from, say, milk that is unable to be shipped for whatever reason. One of them would be included that the cow is treated with antibiotics, and the FDA requires a holding period for that milk after treatment. And so that milk is often collected by producers and used to fed to the calves. And we are working, have done a study on looking at antibiotic levels in waste milk and the impact of that on AMR shedding in the calves.
Well, thanks again, Joanne. I think we're at the top of the hour, so we'll move on to the next speakers. Appreciate it, and you'll join us for the panel discussion later.
Will do. Thank you.
So the next presentation is by co-presenters. The first is Manan Sharma. He's a research microbiologist with USDA-ARS in Beltsville. He's got his PhD in food science from University of Florida, University of Georgia. His undergraduate work was in Florida.
Hey, Jay.
Yes.
Excuse me. Sorry. I think Jim's going to go first. I'm sorry.
Okay. Well, all right. Well, I'll introduce you first. I'll continue introducing you, and then Jim, and then he can.
All right. Thank you.
Manan's research looks at pre-harvest produce safety issues and their interaction with environmental sustainability. And he also looks at the persistence and detection of AR pathogens in water. Jim Wells is a research microbiologist with USDA-ARS in Clay Center, Nebraska. He looks at how animal management decisions impact gastrointestinal colonization and fecal shedding of foodborne pathogens, and also looks at the factors looking at pathogen persistence in the production environment. So Jim, I guess you're going to go first, and then Manan will take over.
Yes, Jay, you can see the slides, okay?
Yes, I can.
Hey, thank you for the introduction. Manan and I are going to basically cover some work that we've been part of a collaborative project with multiple agencies. And this is integrating environmental design, concept, sampling, analysis approach into a NARMS pilot project for U.S. surface waters. And Jay, our host here, has been an important part of this as well, and so has Kim Cook. So for many of you who may not know, NARMS is the National Antimicrobial Resistance Monitoring System for enteric bacteria. The program is about 26 years old. It was established in 1996 to track AMR in these enteric or intestinal bacteria. Some of the initial bacteria that were tracked were those isolates that come from the CDC from their clinical partners. And then starting in 1997, the USDA implemented a tracking program for bacteria from food animals. This was followed in 2002 when the FDA, who administers the NARMS program, started partnering with a lot of state and regional entities to track bacteria from retail meats. The most recent NARMS program or the strategic plan from 2021 charged NARMS to adopt more of a One Health approach. And Dr. Cook talked about this early on in her introductions about how, as agencies were moving more towards this, and it's just not targeting a single specific entity like the people, the plants, the animals, but looking at everything in its entirety and the shared environment in which all organisms are housed. And the expanded tested was suggested to include animal pathogens, and this was, the FDA would be partnering with some of their in-house labs, possibly partnering with USDA-ARS. But on the environmental side, the identified partner was going to be the U.S. Environmental Protection Agency, and the idea was that water samples would be the target of interest. And water samples, just to give you an idea, fairly briefly, water, the surface water, on Earth, everything ends up in the surface water, be it antimicrobials or pathogens from production animals, wildlife, human, pets, through the urban lands, croplands, forest lands, pasture lands. All of that eventually ends up in surface water. And of course, that surface water is being consumed at some point in time, and that redistributes anything that's in the surface water back out. So surface water sampling seemed to be, from the One Health approach, a very important component to One Health. So we were charged, various people were identified from the leadership team and to work on this NARM surface water pilot study. And the pilot study was going to, had to be something where it was a national scale quantitative assessment of antimicrobial resistance within surface waters. And there were various objectives. I'm not going to discuss these at this point. I'm going to come back to these a little later, but there were various objectives or goals that were relayed with this charge. The environmental working groups that were identified, and Jay and Kim were very important in establishing this, there was a design area and a laboratory analytical area. And within design area, there were various groups, including statistical design, in-use, and metadata, in which there was a lot of communication amongst these groups for the statistical design. And then in the laboratory analytical area, field sampling, culturing, and all of those. And Manon will talk about the work of the culturing subgroup after I finish my part. To start off with, I was part of one of the groups I was a part of was the statistical design subgroup. And this was co-led by both myself and Mark Bagley, who works with the EPA. Mark was a great partner in this and very knowledgeable about a lot of the water systems and water studies that were going on. But in addition, there were individuals, active participants from both the FDA, EPA, other colleagues within USDA, as well as the CDC, who made contributions to this statistical design that I'm going to briefly speak about today. Now, as a group, we decided that there were various parameters we needed to consider, and this was having input from the metadata and in-use users as well. And that would include what's the potential for naturally occurring AMR, that background levels that's just in environment, indigenous to a lot of bacteria. We'd also should consider land uses and topologies for the surface waters coming from, soil types, chemistries, applications of lands, hydrology, water chemistries. These are various parameters that we thought about when we were looking at how we were going to design the study, and then also the human and animal sources and how each of those impacts the transmission of AMR to water. So the goals and studies, I'll repeat this, is a national scale quantitative assessment of AMR's surface water. The objectives was to have standardized measures, that something that we would be consistent from year to year and potentially establish consistency from project to project and from country to country. Also, input, we would like to know what are the inputs into the surface water so that we can model AMR risk, help quantify drivers of occurrence and selective pressures for potential amplification, and identify critical control points as means to mitigate where AMR may be introduced into the environment, particularly into surface water. So the goals or objectives were very, very broad, and it was very clear to us on the design group that there was not going to be a single study or approach that would accomplish all of these objectives. So with that in mind, we developed, what we considered a combination of nationalistic assessments and the potential for using intensive watershed studies to answer some of these other goals. We proposed, we thought about a phased design, and this is what, we had worked on, which we would be doing this project in phases for this pilot project. Phase 1 would be initial testing and methodologies. Phase 2 would be validating those methodologies and doing so in a watershed-based assessment. But when we do the watershed study, we wanted to establish something, a demonstration project for others to use for future watershed studies. And then phase 3 would be the probabilistic national survey and looking to see how distributed AMR is across surface waters in the US. When we were looking at this, instead of reinventing the wheel, we looked, are there entities that we could partner with where there are projects already going on? And I've listed some of the relevant US water monitoring programs that we considered. National Science Foundation has some, USGS has their NACWA program. The Army Corps of Engineers monitors a lot of the reservoir water quality during the summer months. And then most importantly, and this is why EPA was identified as a partner, was the National Aquatic Resource Surveys, or NARS. It's a program that's been going on for 26 years, or for 16 years, and it's on a 5-year cycle. And 1 year they'll look at coastal conditions, another year lake conditions, another year wetland conditions. And then 2 of those 5 years, they'll look at national rivers and stream survey. And that seemed to be something that we could use to fill that phase 3 option, looking at the assessing the distribution of AMR and the type of AMR and pathogens across the United States. For the watershed programs, we looked at SEEP that is managed within USDA. National Science Foundation also had, their LTIR program that we could, that was a potential partner, as well as some of the USDA basin scale and aqua sites. In the end, we ended up identifying, once again, a program within the EPA and the East Fork Little Miami Watershed Cooperative. And this has been a long-term assessment of the East Fork watershed just outside Cincinnati, near where the EPA's research labs are. And we will discuss each of these very briefly in the coming slides. So NRSA, or the National River and Stream Survey, it's a national probabilistic survey based on rivers and streams, preliminary study from 2004, first full study 2008 and 9. It determined the extent to which rivers and streams support a healthy biological condition. And they just don't collect water samples. The EPA goes in and does a biological, chemical, and physical assessment of those sites as well, and what's in the water, not just what is in that stream, not just a water sample itself. Every 2 years, over 1,800 samples are taken, and 10% of those replicated across each of the 2 years. Sites are distributed across 9 ecoregions, and there is a goal to sample at least 2 rivers and streams per state. One caveat is that the sample sites have to have flowing water when they're collected, and this is during the summer months. That's also one of the considerations that we thought about, a limitation to this, to identifying interventions and such, is that we would only have a single season in which samples would be collected when they would be collected. So that made it important to also consider a watershed study, something that would be a lot more aggressive and a lot more intensive within a region. And within the watershed, we want minimum measurements and reporting guidelines. We wanted to characterize spatial and temporal patterns of AMR, assess indicators of human, agricultural, wildlife, or natural sources of AMR, and also assess, on a watershed scale dynamics, the attenuation of AMR from point and non-point sources. That EPA East Fork Little Miami watershed study that we identified as a potential program that we could build upon. This is a long-term program. It's a mixed-use watershed, has both human and ag contributions. Those ag contributions, unfortunately, don't include a lot of animal agriculture. There's a lot of cropland contributions and a lot of rural contributions. This site has been studied since the 1980s. The East Fork Watershed Cooperative started in 2001. They have nutrient inputs mapped throughout this watershed. This includes land use characterizations, wastewater treatment plants are monitored, and then the septic systems in a rural environment have also been mapped. And there would be a good capability to determine how those systems, from a human standpoint, are contributing to AMR within the site. Site monitoring includes 29 stream sites. Currently, we are planning to add a few more relative to reference sites and wastewater treatment plants. And there's some lake sites for human recreation. That was an important component as well. And one other thing about this study, instead of just sampling quarterly or monthly, the EPA has established a system where they go out on a weekly basis to a third of the sites. So there's a 3-week cycle throughout this watershed where they're collecting sites, and there are also sites that are base sites that are sampled continuously or a weekly basis, in which they can reference back to for a week-to-week standpoint. So this has led us to the phase design for the pilot project, the initial testing methodologies. A lot of that's been completed. The phase 2, working with East Fork Watershed, is starting this month. And then in 2023, next year, would be the probabilistic survey. And the goal is that if this is successful, in phase 4, this could be a continuation of the NARMS program, looking at both the nationalistic and developing, identifying partners within the country for watershed studies. Now, I'm not going to briefly talk about the everything here. I'm going to stop my screen share and turn it over to Manan.
So thanks so much for that overview, Jim. That's a really nice encapsulation of why this is such an important study and how many different aspects go into this. And as Jim mentioned, you can't really accomplish any one of those. You can accomplish individual factors of those studies, but you can't be as comprehensive if you're not working in a really interdisciplinary group among different federal agencies. So it's been a really rewarding effort to be a part of. And so we're going to talk a little more about the culture methods that Jim mentioned and that are part of phase 1 of this study. And really our objectives in this part were to be able to compare Salmonella, E. coli, and Enterococcus recovery and filtration methods from surface waters. And this is just the first part. We know we're talking about antimicrobial resistant pathogens here, but in this case, we need to identify the organisms and recover the organisms first before we do any of the antimicrobial characterization. So this is what my talk is going to focus on for the next few minutes. And so our objectives are really to identify methods to be used in a pre-pilot watershed study and all the different aspects that Jim mentioned, and determine if those methods can really be integrated into these assessments in a practical manner. And in our case, the practical manner is basically, how does this work in this integrated program? And this is taking into account very sort of on the ground things like how much time does the sampling team have? What kind of resources does it have? How much water are they able to carry back? Is it better for them to filter this water in the field or in the laboratory? What are these sort of logistical concerns and sort of cost-effective ways that we can get be the most efficient and get the most sort of microbial data from the methods that we choose to employ? And that's what we really wanted to evaluate in this study. And so our objectives really were to determine the presence or absence of certain pathogens or indicators, and in some cases, the quantitative recovery. So for Salmonella, we were only looking at the presence absence in our water samples. For E. coli, we wanted to look at both the presence absence and the quantitative recovery of selected AMR phenotypes, and then specifically determine which methods would be better at recovering Salmonella. Can we use standard inoculum to evaluate these? And we'll talk about that more in just in a little bit. And what scheme can we use? Like, how can we, again, get this scheme to fit into what is going on with everything else that's going on in these large surveys? So we picked 4 different methods for our Salmonella work, and I'll talk about our Salmonella work a little more extensively. And we had 4 different methods. We wanted to try and do a bulk water method. And this is sort of the no frills method, the really cheap method. You just get water, you add some microbial media to it. It's very low effort. You just add something, you incubate it, and then you go through a Salmonella enrichment and isolation process. And this is, again, just to save time and money to see how that would work. We wanted to evaluate a modified Moore swab or a vertical modified Moore swab. Modified Moore swabs are basically rolled up pieces of cheesecloth. You can put them in a PVC container here. You can pump a lot of water through them. It's been commonly used in a lot of different studies evaluating surface waters over decades. So that's why it's an attractive option here. For the modified standard method, we wanted to basically filter volumes of water through a glass wool filter and incorporate some pull filter, which is a cellulose powder, into that water and collect the bacteria on the filter here, and then again, put it through our Salmonella isolation procedures. And then finally, the dead-end ultrafiltration method. And this is a method that's based on our FDA colleagues in something called the BAM, the Bacteriological Analytical Manual, where they look for a parasite called Cyclospora. And this has been a really effective method for them to do that. And the CDC has also used this in foodborne outbreak investigations, where they're trying to isolate pathogens like E. coli 0157H7, which are really have a low prevalence in water, and it's really effective in doing that. It's a high volume filter, and it's usually a wet dialyzer that's used in kidney dialysis patients. So just a little bit about what we did. We collected water from a site here close to us in Maryland. We shipped this water out to our colleagues at other ARS locations. We took that water and inoculated it with something called a Salmonella typhimurium bioball, which has a green fluorescent protein marker in it. All the laboratories did this on their own, and then ran this through a Salmonella scheme. And this gave us a very low inoculum in our water. Those bioballs are about 30 CFUs, so we're adding 30 CFUs to a liter, giving us a pretty sensitive assay. So if you recovered it, you could recover at least 30 CFUs from 1 liter of water. And in this first part of the Salmonella phase, we were evaluating the bulk water method, the more modified more swab method, and the modified standard method here. So our protocols that we developed, and we spent a lot of time talking about these protocols and developing them in the lab and in discussions with the interdisciplinary group that Jim mentioned. We have posted all these protocols on a website called protocols.io, because again, our intention is to hand this off to our EPA colleagues who are going to do this watershed assessment that's beginning this month for the pilot study. So all of our protocols are here. They're transparent. This way you're not relying on somebody emailing you or somebody's Word document getting corrupted or somebody not showing up for a Zoom call. So we've tried to be pretty transparent with what we've been doing with our methods here. And we really want to thank Kim Cook and Betty McCon for facilitating this and giving us a platform to do that with. So going on to a little bit of our results for part one, what we found was a couple of things that when we were comparing those three methods I just discussed, we found that the modified standard method recovered Salmonella typhimurium more frequently than the modified more swab method or the bulk water, and that's across all laboratories. And then you can see these red and black circles here. So we had some laboratories that showed more proficiency with some methods than others. And that's pretty much to be expected if you're shipping it out to 4 different laboratories and you're all doing the same thing. You're going to have some lab to lab variation, but that was good for us to see because that really emphasized to us the need for sort of training and getting people familiar with the protocols and things like that. So this wasn't really unexpected. But what this did tell us is that even with the labs that were having a lower frequency of recovery, still the standard method was performing better in those laboratories, and the labs that were recovering Salmonella more frequently, the standard method was doing as well as other methods. So this led us to identify the standard method as one of the methods we wanted to use. We then compare that to the dead-end ultrafiltration method in a study that just involved 2 different sites, ARS sites, using the same Salmonella bioball. And what we found here is that the modified standard method and the dead-end ultrafiltration method were roughly equivalent in their frequency to recover Salmonella. And I think for the use in our study, I think what our preliminary results tell us now is that the standard method is more suitable for this NARMS pilot project, since the filtration is easier to perform in the lab than the dead-end ultrafiltration. It's just a little easier to do. You can get more high throughput, less setup, and you can go through a lot of samples more quickly. So in this is how we identified the standard method for the recovery of Salmonella. Our colleague, Lisa Durso, has spent a lot of time developing methods for the quantitative recovery of E. coli from surface water. And she's put together a really nice set of protocols that we have all used in a similar way to evaluate these E. coli methods. So they're developing, they're including membrane filtration-based methods and a most probable number-based method, which is the CoLalert Quantitre assay. And we evaluated those methods using the same laboratories I showed on a previous slide, evaluating these methods separately, using different types of media, using pan-sensitive E. coli as a control, and also multi-resistant E. coli, resistant to tetracycline and cefaxetine. And this is a really nice study. We've shown that you can recover really low levels of E. coli and the multi-drug resistant E. coli. And Lisa's set up and done a lot of work to set up agreements with commercial companies to sort of produce a preparation of these validated strains that can be used by anybody in the way that we did it. So a lot of work was done here for the E. coli methods as well. So really, our conclusion so far is that we can recover low levels of Salmonella using the modified standard method. And we feel like through our evaluations, this is the most suitable method for some of the pilot studies that Jim mentioned that are coming up. They're going to be starting this month. And really, it's the most suitable method because it can be incorporated into the logistical and laboratory capabilities that we've learned about through all of our meetings and discussions through these various groups. And it's really easy to incorporate with the shipping and filtration of water in the laboratory from the field crews to back to the lab, things like this. And the quantitative methods for the evaluation of antibiotic-resistant E. coli were developed, and we also feel really good about the reliable recovery for these set of methods. I think this study will use a slightly different method when in the pilot study, but I think these methods are going to be useful to a lot of people in the future. And finally, just this sort of basic, but the laboratory proficiency with all these methods can be developed in a short period of time. Our lab learned how to use a lot of them. I know other labs have not used them before, but we learned how to use them as well. And that's a really good sign for the larger national study where you may have a laboratory team that needs to be trained and can use this. So with that, again, Jim Wells mentioned all of our collaborators, our USDA-ARS collaborators have been really good about doing the laboratory portion of this work. We've gotten so much good input from our EPA and FDA collaborators who've advised us on filtration and laboratory methods. So we just really appreciate everybody's input to this point, and the interagency agreements we've had between the FDA and USDA and our ARS funding. So thank you all very much.
Thanks a lot, Manon and Jim. Great job. Part of the problem with having two speakers for one talk is that I think we've kind of run out of time for questions right now, but both of you will be around for the panel discussion, so you'll be available then for questions. There was one online real quickly. They asked about the Salmonella bioballs. Is that a commercial product or is it something your lab developed?
That is a commercial product made by bioMérieux. We wanted to have a sort of a standard inoculum, and we thought it was just easier for every lab to be able to order this and know that we would have the range of between 28 and 34 CFU per ball when we were adding it to our 1 liter samples. So,
all right, thanks. All right, so we'll turn to the final speaker before our panel discussion, and that's Danielle LeMay. She's a research scientist with USDA-ARS in the Western Human Nutrition Research Center. Danielle got her PhD in nutritional biology from UC Davis, and she also got her undergraduate in BS in electrical engineering and computer science from MIT. Her research evaluates biotransformation or bioinformatics to study how dietary components affects host response and whether that response is mediated by the resident microbiome. So Danielle, I'll turn it over to you. And I can see your slides.
Great. Can you hear me?
Yes, I can hear you too.
Wonderful. Thank you very much, Dr. Garland, for that introduction. And thank you to the organizers for inviting me to speak with you today about the association of diet and antimicrobial resistance genes in healthy humans. So I probably don't need to convince this audience that antimicrobial resistance is a problem, but I'm speaking with you today about a project in human health. And so I just wanted to bring to light that it's a growing global health threat to humans, and projected that by 2050, AMR will be, killing 20 million people per year. Hopefully with the, USDA-ARS and other entities around the world working on this, we can change that projection. So I'm a nutrition scientist and am coming to AMR from that perspective. What do we know about diet and antimicrobial resistance? So I've done a lot of work in the milk biology area, as has my colleagues, and David Mills's lab has pioneered looking at AMR in early life in humans, and looking at whether infants are consuming breast milk or formula. We find that infants that are consuming breast milk, they have their microbiome, their gut microbiomes become completely dominated by bifidobacteria, and that's because these bifidobacteria species contain cassettes of genes that allow them to consume the oligosaccharides that are in milk. And so these species are really milk-oriented microbes. And it turns out that bifidobacteria are pretty resistant to acquiring antimicrobial resistant genes. They don't acquire them quite as readily as other taxa. So breastfeeding infants have much lower AMR in their microbiomes, but this is not sustained in the long term, meaning after they've weaned and gone on to solid foods. We have also looked at the fecal resistance of dairy calves. So similar situation, but in cows. The cows are, initially fed a colostrum. This colostrum doesn't come straight from due to animal management practices. The colostrum doesn't come, straight from the mother cow. It's collected from the herd, and so we had sequenced both the colostrum and the fecal metagenomes from these dairy calves, and found very similar patterns of AMR, suggesting that the colostrum itself was seeding the antimicrobial resistance. Now, it turned out that as the dairy calves were weaned, that in their diets, of course, have increasing fiber, that AMR decreases over time, which is a good thing. So these two studies sort of gave us a sense that diet may have an impact on antimicrobial resistance, both influencing the decrease of antimicrobial resistance, or even possibly being a source of antimicrobial resistance. There are other, so looking towards adults, there have been observational studies showing that industrialized populations harbor more antibiotic resistant strains. So for this study that was published in Science, they had looked at the microbiomes of hunter-gatherers, and then comparing that to microbiomes of people in industrialized nations, and those of us in the industrialized nations have more antibiotic resistant strains. Now, this is, of course, like lumping all of us together, and we are not a monolith. We eat different things, and so we really need to understand, do our different dietary patterns matter? So that was the impetus for this current study of determining the association of diet with antimicrobial resistant genes in healthy US adults. So we leveraged the USDA nutritional phenotyping study cohort for this study. That's a study of healthy adults aged 18 to 65. Both men and women, and recruited in different BMI ranges of normal, overweight, and even moderately obese. And their dietary intake was monitored using two different dietary instruments. The Block Food Frequency Questionnaire, or FFQ, was used to ask about habitual diet over the previous year. And we used 24-hour recalls, automated self-administered 24-hour recalls, to measure recent dietary intake over the 10 days leading into the stool collection. We sequenced metagenomes from 290 individuals in this study. Before sequencing those metagenomes, we had used publicly available data and even simulated some metagenomes to determine what parameters we needed for sequencing. and found we needed 150 base pairs or better. And key for human metagenomes, at least, is we need to really deeply sequence the samples to make sure we're able to quantify those low abundance AMR genes. We also manipulated the library prep to enable overlapping reads for gene-centric analysis. And then there's the bioinformatics analysis that took quite a long time to put together the whole pipeline and run all those samples through the pipeline. And I won't drag you through this, but I'd be happy to take any questions if people have questions about the methods. So on to the more exciting part, the results. So as you've seen today in cows, and this seems pretty common across mammals, that aminoglycosides are the most prevalent in this cohort of healthy US adults. So the aminoglycosides are this dark blue here. So the second most common mechanism is MLS, followed by tetracyclines. So one of the more striking things is noticing how AMR is pretty variable across all of our individuals here, that they're stacked in order of abundance. So we divided up our data set into the quartile, the low AMR. So that's the lowest quartile of gene abundance, and then most people here are in the middle, and then the high AMR cluster is the upper quartile. So what about diet? So our primary hypothesis was that increased dietary fiber intake would be associated with reduced antimicrobial resistance. And we had some secondary hypotheses that the healthy eating index, which is an index of diet quality, would be negatively associated with ARG abundance. We expected habitual animal protein intake and habitual saturated fat intake, which is also kind of indirectly related to animal protein intake, would be positively associated with ARG abundance. And finally, we expected stool pH, which is an indirect marker of fermentation of fiber in the colon, would be positively associated with ARG abundance. Okay, so we directly tested these hypotheses, and some hypotheses have multiple variables associated with them. And we found that in the case of fiber, whether it was from the ASA24, as a reminder, the 24-hour recall, or whether fiber was from the food frequency questionnaire, we were finding that it was highest in the low AMR group. And habitual intake from animals, particularly beef and pork, were lowest in the high AMR group. So having answered our direct hypotheses, we wanted to, this is a really rich data set. There's a lot of data associated with this cohort of humans. So we wanted to use machine learning to really rake through all the possible variables to identify any other possible predictors of antimicrobial resistance. And so, we built machine learning classifiers to take all the data and then classify the individual as high or medium or low AMR, and then used Shapley additive explanations to explain the magnitude and directionality of those features. I should say that we're using machine learning in a strange way here. Our goal isn't necessarily the prediction so much as the explanation. So machine learning is only as good as the features you put into it. And so we spent some time trying to really come up with some additional good features. And it's been, for those of you who are microbiologists, that, microbiology has a natural taxonomy, and this taxonomy of microbes really useful information. You can use tree-based metrics to measure things like diversity. And food also has a taxonomy, not as well annotated as microbes do, but it has been shown pretty recently that we can put food into something of a hierarchical tree that we can then use to apply diversity metrics. So before I dive into the machine learning results, I just want to explain how to interpret what these graphs look like. So Shapley is this mechanism this algorithm developed from game theory to help determine which features are important. So a more negative SHAP value here would predict lower AMR. More positive SHAP value here would predict higher, well, in this case, the medium AMR group. And then the color here is indicating like the value of the feature, whether it's high or low. So for example, the phylogenetic diversity of all foods with a higher value of diversity, so higher diversity of foods, is more predictive of low AMR. And likewise, a lower value of dietary diversity, more predictive of medium AMR. Okay, so now that I've explained what this is, we'll go through the rest of them. So yeah, so here, higher diversity, lower AMR. Here, higher DHEAS is more predictive of medium AMR. And likewise, higher epinephrine also associated with medium AMR. And then soluble fiber and total fiber, you can see the magnitude is not anything as high as the diversity of foods, but higher soluble fiber or total fiber was associated with or more predictive of low AMR. So we built models to distinguish all three clusters, high, low, medium, or low, medium, or low versus high. And the models really were not able to predict high AMR very well. We had the best results with distinguishing low from medium, but not low from high. And our way of interpreting this is that that the dietary and lifestyle measures were really not capturing what drives individuals to have the high AMR, which may be something like antibiotic usage. So what I've discussed so far, we did not include microbial features in those models. Now, when we bring microbial features into the model, these much more accurately, of course, predict AMR, as some taxa are known to be more associated with AMR. So as you can see here, Enterobacteriaceae and Streptococciaceae were higher in the high AMR group. And conversely, strict anaerobes like Clostridiaceae species were higher in the low AMR. These are strict anaerobes. So in conclusion, higher fiber intake and more diverse diets appear to be predictive of a lower antimicrobial resistance gene abundance. Individuals with lower animal protein intake have lower antimicrobial resistance gene abundance. And finally, high AMR is associated with high Enterobacteriaceae abundance. And this is actually a tree of all the different, the taxonomy of the different diets that people were consuming. So for this project, I'd just like to acknowledge MVPs from our lab, the postdoc, Dr. Andrew Oliver, who did all of our wonderful graphics and analyses, and Dr. Zayas Yue and Yuri Tang, who had both worked on the bioinformatics pipeline. And of course, the entire USDA nutritional phenotyping study team that produced all of the data. So with that, I'd be happy to take any questions you may have.
Thanks a lot, Danielle. It's very fascinating. The complexity of the consumer, right, is to put in the context of the overall food production system. Do you mind asking or kind of discussing your original hypotheses about, say, high fiber being negatively correlated with AMR? Do you want to go in a little bit about the rationale for some of those original hypotheses?
Sure. Thanks for that opportunity. So the rationale for that hypothesis was really from that dairy cow study where we saw that the dairy calves. So in that particular study, we had very frequent sampling, and had saw that for the dairy calves that were put on the high, as they were increasing the fiber of their diet, the AMR was reducing. And so that's where this hypothesis came from.
Okay. And if I'm reading your results right, it speaks to, well, eating a diversity of foods is definitely a positive impact.
Yeah.
And eating higher fiber is a positive impact. Does that mean we should just stop eating meat?
So great question. So I would say no. So first of all, this data was observational, and as you know, it's all correlative. And one of the complications is, I mean, people eat dietary patterns, and so the people in our group who were consuming the high fiber and the diverse diets happen to also be consuming less meat. But so then it becomes tricky. Which one is the driver? Is it the higher fiber or the less meat? But the higher fiber had a far stronger signal in our data than the less meat, and so we saw that with both the machine learning and in our direct hypotheses. So that leads us to think it's the diversity of fiber that's really driving. The reason, dropping back to first principles, fiber from different sources is going to be providing all kinds of carbon sources to the microbes. So when we eat, we're not only feeding ourselves, we're feeding all our microbes. And when you're providing a very diverse set of carbon sources, that's going to create a more diverse set of microbes and cover all the different environmental niches and make it more resistant to pathogens and so forth. So it should lead to a healthier gut. The second thing I want to say about should we stop eating meat? There are nutrients that are really hard to get with plant sources alone, and animal sources provide all those nutrients, and they provide them more inexpensively. And so the other caution is nobody in our cohort was a vegan, so we just simply cannot say stop eating animal sources. So yeah, thank you for that question.
A question from the audience is, are there links or hints from the consumption of more processed foods and AMR?
So that is also a great question. And unfortunately, we don't have the resolution of the dietary data that we need to answer that question. And there's even, it's kind of an open research question, even how to classify processed foods. There's like a NOVA classification system for how processed a food is, but that doesn't really track with how healthy a food is. So for example, yogurt would be classified as a highly processed food, but it might be one of the most nutritive foods you could eat. So it's really, it becomes very difficult to do that type of analysis. Yeah.
I think we've got a couple other questions. On the fiber and meat correlation, is there a plan and interest in follow-up to the study to vary the studies of subjects based on their diet?
So that's a great question. I do think that we absolutely need to do an intervention study where we have people on controlled diets and can directly manipulate the amount of fiber, the sources of fiber, the amount of meat, the sources of meat. Absolutely, that's the way to be approaching this in the future.
And kind of a related question too is, does this mean that gut microflora keep changing with the food that we eat?
Yes,
Yeah.
yes, they do.
It's a very dynamic system. All right, well, I think that's all the questions that are online. And I guess the goal here is to, we heard a range of specific talks on kind of, like I said, the broad complexity of both detecting AMR in the food production systems, but also kind of understanding the AMR in the context of the drivers of the occurrence. So we can, instead of talking specifically about the presentations, it's really try to get these broader goals. The ARS effort here on this series of webinars, this one on systems biology and detection, what they're really interested in is kind of understanding both what are the gaps and the needs for improved detection, and how do we translate that detection into improved decision support tools, risk models, so that can be applied to really try to mitigate the risk in the context of food production. So kind of thinking a little bit more broadly, kind of like to get the panel to think about that and discuss it. And I guess I'd just start off, open question for all of you panel members. What do you see as, and I know this is a difficult one to answer because it's a pretty broad, vague question, but what do you think the gaps are, the biggest gaps are to informing decisions about mitigating the risk? Is there anything that stands out to you as a real key gap? And I guess I see Danielle, you're up on screen. You mind giving your thoughts first?
Sure. So first I'll speak a little to the human perspective, and then food system. So from the human perspective, like I mentioned, like we've only done observational trials so far, meaning we've only like observed people in the wild or free living where they're eating whatever they're eating, and those tend to fall into certain patterns, and so it's very difficult from a scientific perspective to separate out the variables and understand the what's driving either AMR, or it may be that AMR is actually coming from some source that's not food at all, but then our food influences that resistome over time, whether we harbor that reservoir or suppress it. So we absolutely need intervention studies in humans, and then secondly, like in addition to food, like, do we need to be providing probiotics? Do we need to provide symbiotics? So a symbiotic is a probiotic together with the prebiotic that's specially formulated for that probiotic. And so, that's where, we directly feed the microbes and the food for the microbes. And maybe that is a solution in humans. It may also be a solution in animal production systems. And I think having these kinds of webinars that go across the food system is really great, because we human nutrition scientists can learn a lot from the animal scientists, and vice versa, through that comparative biology. And then across the food system, we do need to be doing some more sampling to understand, are there sources of AMR in food, and how does processing affect them, and so forth. I think I suspect that AMR is not a huge issue with food. We did specifically look at retail milk, and found that all the pasteurized milks were not, there was very low AMR. There's just very little bacteria, and we were looking at HTST milk. Of course, in ultra pasteurized milk, the bacteria is completely killed. So the only source of AMR we found was actually in retail raw milks. A lot of the country you can't buy raw milk, but out here in California, there is raw milk right on the shelves, and so people can buy raw milk right at their grocery store. And we found that even though the raw milk didn't have specific pathogens that we were testing for, they were contained, lots of antimicrobial resistant genes. And when you see the data, you're like, oh, I would not want to drink that. So there's actually a different public health issue with drinking the raw milk. So I suspect that sources of AMR in food are rare and going to be those particular niche products where there's a lot of microbes in the products. So maybe I should stop talking and let others have their go at this.
I guess, Joanne, since Danielle kind of brought up milk, I don't know, anything you want to add on that specifically, or just the general question of what do you think the key gaps are for data to inform decisions?
Well, I support Danielle's comment about, raw milk being a concern for a number of reasons. But one of the things that I think we really need to try and understand is we have a lot of data on AMR in food production systems and animal production systems, and we have a lot of sort of prevalence data. Is it there? Is it not there? How much is there? What genes are there? Are they an issue? Which E. coli? How many resistant E. coli? As some of the data I showed today, and so we know if we have a resistant Salmonella typhimurium in the system, that's not good, right? We recognize that. But what about what's the risk? What are we really concerned about the other like the non-pathogenic bacteria in the system that are resistant, or this plethora of ARG that we find, resistance genes in the system? I'm not saying we shouldn't be concerned, just saying, there's so much information there on what's there. I think we need to start to tease out, like, how do they distribute throughout the whole system? And as Jim and Manon had pointed out in some of their work, there's, we have to consider the environment, the crops, the animals, the food. And so, that's what I try to get my head around is, like, what matters? What's the most critical? Certainly specific pathogen, the pathogens are of a concern, and certainly specific combinations of drug-resistant pathogens are a concern. We see a lot of salmonella, like in the experiment I showed, we see a lot of salmonella in these systems that, for example, are pan-susceptible all the time when they're exposed to resistance in that animal, in that gut or feces. So I'm kind of rambling here, but I guess where I get back to is to move forward, we have to start to identify some of the more like, so where do we need to focus, and what are the ARGs that we're concerned about, and the locations. And to get that, we need much more information on how do they move through the system, in my opinion, through the whole system.
Now, I do think your work was one of kind of clear indication, or a really good example of trying to look at the drivers for the occurrence, right? Which is, you're talking about, trying to understand, what's the role and kind of the origins of a lot of the non-pathogenic AMR, or AMR not associated with the pathogens in the system. I get that, and that's the complexity. But what I thought was really powerful about your work was that, okay, we see all this, right? We see the occurrence, but what's really driving it? And I think you've got some very strong hypotheses about what's driving it that some targeted mitigation work can try to resolve.
Well, and yes, and then our question will be, like, when we find these targets, for example, if we are able to, it's probably not as simple as this, but if we can add iron to the milk and to the colostrum and have a significant reduction in AMR in the calf gut, does that translate? Because the AMR, there's still AMR as the animals age, the percentage of the E. coli population and the and the total ARGs in the feces reduces as the animals age, but it's still there, and they're still there to be selected for under antibiotic treatment. Now, if we can reduce the levels of especially these highly resistant bacteria in the calf gut, we have to follow up. Does that translate to as the calves age, will we also see these reductions in the other animals as they age, and in the animals in the adult animals on the farm, and then in the system, and in the products and in the environment?
Right. So, Jim, I think, as someone who, as your talk indicated, spent a lot of time trying to understand or try to look at the statistical design for what is kind of an observational study, right? I mean, it's a monitoring effort, but trying to design it in a way to inform some of the drivers. Any thoughts on, because we talked about some of those issues already. I mean, what, in your mind, from the work you've done, what do you think the gaps are in terms of really developing more mechanistic understanding of the environmental component, I guess, or your other work, just looking at food production systems in general?
I think, the starting with that animal management, there's been a lot of work, and Joanna's worked a lot with the dairy cattle. And, from this data that we've got coming out now, I think there's, she mentioned there's still a lot more work we need to do to see what could be some of the drivers within those systems. From my perspective, that's the case for both beef and swine. We know there's antibiotic resistance there. We still don't know what's driving a lot of the diversity and the changes and the shifts in those populations is leading to antibiotic resistance. And, and once we figure out what could be then we can do like Joanna's done. It's like, maybe it's iron binding proteins or things like that, or the iron sequestering systems that's leading to these E. coli populations to be, that have antibiotic resistance being more populated in the animal guts. But then, also considering, what happens with the feces and manures and what happens in that environment. And beef cattle production, those environments are all open. Now in CAFOs, a lot of that nutrient and those bacteria are all sequestered within lagoons and stuff. But at some point in time, that does get applied to the land, and what could be happening with that. And that, or even, if there is some cycle where it goes back into the crops and back into the animals that consume those crops, and then also into the water systems. And then once we have some information about the water systems, how does that come back in, to the cropland, and also maybe even to some of the animal populations themselves, or even wildlife that could carry it back into those production animals that we have in open systems. So there, from my perspective, we still have a lot, particularly in this One Health environment, a lot of questions we need to consider, and we need to develop studies to resolve those issues.
Yeah, and I mean, I think that's been one of our challenges within the ARMS program is it's observational, but we're trying to develop this monitoring or surveillance program in a way that can inform some of these drivers. And that's why, like you mentioned, you came up with this phased approach, looking at, I'd kind of call it going big and slow, right? Trying to look at the whole national picture and try to understand that, but knowing that that's kind of a slow process or not high frequency sampling. You also need this more intensive watershed sampling. There was a question Q&A about the NARMS program, the water program, and it was, is there metagenomics work? And there is. I mean, what Manon talked about was the culture-based work, but not just for the work that we're doing in NARMS, but also other kind of method development work for water testing for AMR. It involves kind of a 3 levels of analytical approaches. One is culture-based approaches, looking at resistant pathogens of concern. The second is, and which is great, that translates very easily into risk, and that's the kind of data that the human side of the One Health continuum really wants to see, as well as the animal health too, and that's why we're looking at salmonella, because of its prevalence in the food production systems. But it is a fairly limited view, and it's labor intensive to do that. So we also want to look more broadly at a broader range of resistant gene targets that are there. So we're doing that as well as a broader metagenomic approach on the water samples. So kind of all three different levels of analysis. That's the plan to do on the samples that the studies that Jim mentioned. The other kind of side of this is, we're talking about, how do we use the data? But the other issue is, what are the gaps in technologies for more rapid detection? I know Walt's not on, but in his talk, he mentioned about trying to put technologies in the hands of the growers, right? Where they could be more rapid online detection or more real-time detection of the risks in the field. The question is, what do we think the real key gaps are there in the technology side of things? And Manon, I know you're on the culture side, but as someone who does a lot of measurements, I'll kind of start with you on your thoughts on key gaps in the technologies for more rapid detection. And I guess maybe, the assumption there is that more rapid is faster is better. Maybe, I'd hear your thoughts on what do you think gaps are for more effective monitoring, I guess.
Yeah, thanks, Jay. I think a lot of us associate what we call culture independent testing as with more rapid, and that's a good point that you make. I don't know about it. Probably can be more rapid. Is it as accurate? Does it tell us what we want to know? We've got some data lying on my desk in collaboration where we've taken water samples where we get a culture positive of salmonella, and then we've tried to take what we call the enrichment, the non-selective enrichment, where everything grows up, and run a PCR on that enrichment and see if we can fish out the salmonella and other pathogens of interest. It's really not a straightforward relationship, because as Jim mentioned, water is sort of a repository for everything. There's so many things that can interfere with a PCR reaction. I do think our FDA colleagues have been, and Andrea Otteson of the FDA has been leading this with what she calls a quasi metagenomic approach, which is where you don't take the water sample all the way through to the culture sort of enrichment and isolation. You enrich it, and then you look for specific targets. And she's obviously been focused on antibiotic resistant genes, but that platform could lead to a lot of other detection of other, say, like pathogens of interest or specific ARGs or other genes of interest that could be markers for pollutants or pathogens. So I think that approach may be promising, but it's also expensive and labor intensive. It's not as easy to automate sometimes. And I think one of the things that would be really interesting to do, and this is why I think the pilot programs that we've discussed and worked on for the last 2 years, getting all of these approaches together, is that, we're ARS, we focus on a lot of agricultural environments, but I think the more you read about water and you realize that a lot of the antibiotic resistant gene burden or antibiotic resistant organism burden in water may be related to humans, and how you map these mixed use watersheds with septic tanks that may be failing because they're old or they haven't been well kept. And I think markers that could differentiate, I know we have a lot of microbial source tracking resources. I'm not as familiar with them, but I know that they could probably be integrated into something like this, into sort of a PCR suite or something where you can differentiate where the contributors are. And that gets back to the points that Joanne makes about what drives a lot of this. So I'd be really interested to sort of see how all of that could come together. And I think you could probably design some nice molecular assays to figure out what's the human burden, what's maybe the non-human burden, what's the agricultural burden, what's the non-agricultural burden, without implicating, stakeholders or anything like that.
Yeah, no, that's a good point. Water, your strength is your weakness, right? The strength of water is that there's inputs from various sources. So it is a good integrator of the impacts at a watershed scale, but at the same time, that's a weakness because it's complex, right? You really have to be able to tease out these various, like you mentioned, the different factors that are affecting it. And I do think, yeah, fecal source trackers to really relate the occurrence of those to certain kinds of resistance would be useful. And it speaks to what I think what Jim was emphasizing, this need to really understand the relationships, not just the occurrence, right? Really what again, back to this point of understanding the drivers. Anyone else have a comment on kind of tech gaps in technology for detection? I mean, we talked about water a little bit here, but in any of the food production areas of concern.
I think one of the challenges of trying to develop these technologies that are sort of useful, for the water work and for the on-farm work. We, in animal production, with a lot of different matrices, like feces is not easy either to get in there and get the DNA out, and actually the concept for me of doing, we can do this in the laboratory. In the last 10 years, has been incredible development on what we're able to do, and from a standpoint of genomics, metagenomics, and looking at the resistome, but trying to think about getting to a farm side analysis, which is what in many ways we're talking about. I think there's a lot of challenges there, and we need a lot of effort in that area to make it work in different matrices as well, to identify what we're actually looking for, to make it work in the different matrices, and I had another thought there, but to make it also, and get the detection limit down, right? We have that problem with PCR, and Manam was alluding to that, and to actually make it useful at that level. I think we have a long way to go there. The only other thing I wanted to add, if I may, is we probably, and I'm not totally familiar with what's available, but we probably need some more sort of animal side tests for specific, say, drug pathogen combinations that a producer can use to say, to inform their treatment of that animal or that pen or that flock, depending on the situation. And I would think that we could use some more advancement in that arena to help with more effectively using lower amounts of precision antibiotic use, I guess I would call it. Yeah.
I don't know, Jim or Danielle, anything you want to add on kind of the detection side of things and technologies related to detection?
I was wondering how much. I was wondering how much the field of food safety would inform this kind of sampling for AMR, just because food safety has the same issue of, well, you'd love to sample everywhere all the time, but you can't. Right. And you need to find sort of low incidence things. And in the field of food safety, what I see happening, it's not my area, I should say, but I'm tangential to it, and from an AI perspective, I see what's happening. And there's a move towards decision support tools to determine where to sample, how often to sample. And I would think some of the modeling technologies that are being developed for food safety with AI could be applied in this domain of AMR monitoring.
Right. Well, Manan raised his hand. Manan, go ahead.
Yeah, I think Danielle raises like a really good point here about using machine learning to improve sampling and targeted, maybe trying to identify hot spots in a production environment. I think that approach is really, really good when you have a controlled process. When you have, a manufacturing plant or some process that's going through, you are enclosed inside. And the challenge here, I think, is, and we see this because we work in produce safety, when you have fields outside, or you have, say, emerging controlled environment agriculture like hydroponic or aquaponic environments, and you introduce those variables, the sampling plans are still really complicated. And there's just, I think Walt mentioned this in the first talk. I mean, it's basically outside, right? Like, it's really hard to identify the drivers of what is going to promote the incidence of foodborne pathogens in that field. And I think that's one of the data gaps here. You see a lot of attention being paid to this by the produce industry about trying to integrate these machine learning concepts with these big data sets that they're trying to collect so the machine learning algorithms can have enough to churn through. But I think from a produce safety side, it's not there yet. I think from a manufacturing side, where you have that controlled environment and you have a process, I think there's much more immediate potential to do that. And that's definitely an area that we need more resources and more expertise devoted to, is like this produce safety, in agriculture and sampling plans.
And I guess getting back to the question of surface water, when you say more uncontrolled production, like say irrigation or even livestock drinking surface water, I mean, what are the impacts of that exposure pathway? And I guess one thought, and this is something that I know this is food production side, but from the EPA side, one of the things we worry about is, what's risk of ingestion. Probably not from drinking water, but probably more from recreational exposures, where you're just ingesting untreated water during some kind of recreational event. And I maybe this is for Danielle a little bit. I mean, but also Joanne, you raised this question, was, the simple kind of risk question there is, how many of some MDR pathogen are you ingesting? And that's something we're trying to develop data on and develop risk models for that. But the other side of it is, given the dynamics of what goes on in the microbiome, the other concern potentially is just what's the risk of ingestion of the non-pathogenic AMR into your system, and what impact does that have on your resident microbiome? So not necessarily and maybe introduction of the transfer of the resistance into organisms of concern in your gut. So not the direct exposure to the pathogens and the resistant pathogens, but the exposure to the genes themselves, and then secondary impacts from that within the microbiome. And I don't know, Joanne or Danielle, I know somewhat related to your areas. Is that something that is under study?
Yeah. So I won't go so far as to say it's under study, but I'll say what I've observed in healthy humans, in that, so for example, we saw aminoglycosides was the most common mechanism, but when we dove down to look at the taxa who harbored those aminoglycoside resistant genes, it was everything, like all kinds of taxa, and it was just pervasive. So clearly, this is something that all kinds of non-pathogenic organisms have acquired. And so that's okay, because it's, if you have, lactobacilli or bifidobacteria or something that's resistant to antibiotics, well, that's just fine. It's more of an issue of when I've had, if you've got some E. coli that comes through that then, takes that gene and because, so it's more of an issue of specific pathogens acquiring those genes. And so it's a huge question. What matters? Is it the specific pathogens, or do we need to suppress all kinds of antimicrobial resistance? I mean, antimicrobial resistance naturally occurs. If we had never ever administered an antibiotic of any kind, there would be some antimicrobial resistance everywhere. Like it's a naturally occurring phenomenon. But I think we really need to focus on the medically important antibiotics and genes that confer resistance to those most medically important antibiotics.
I guess I certainly agree that we need to maintain a focus on the medically important antibiotic genes or antibiotics genes that confer that resistance, but we have to watch also for cross, ability of this, if we're looking at bacteria that are resistant, or we're feeding, administering certain antibiotics that aren't medically important, there's also the potential for those raising also resistances to medically important antibiotics. I'm not very familiar with that area, but I think that's something we have to keep in mind. But Jay, I think you're very right. I don't think we can answer that question yet. And I think that part of the bigger picture is, this webinar series has been designed in these series of 4, I forget, 4 or 5, but the first one last month was on risk assessment. And so, we sort of all need to work together and try to figure out, the risk assessors and the modelers, they're working on developing models, and I guess, we have to not let them do that in a vacuum and not let us do our work in a vacuum. We have to keep sort of these big conversations going so that we can focus on this question. Maybe there's data out there that can be used to start modeling some of that aspect. What's the risk of ingesting just ARGs that are not associated at this point with any pathogenic bacteria? But looking at the E. coli population in general, when we isolate E. coli from dairy animals, and the same is for beef, and it's not, it's primarily they're primarily non-pathogenic isolates, but of course we're also isolating shigotoxigenic, uropathogenic E. coli, and so there are E. coli there of concern, and as well as the other pathogens that we talk about, salmonella and listeria. And it's just really hard to get your head around, what do you have to test in order to be have a good understanding of what the risk is associated with that system.
Yeah. But I guess what I've heard so far is that number one, we can already collect large volumes of data, right? I mean, so being able to collect that data faster isn't necessarily the key gap here. It's really utilizing and doing more manipulative experiments, like Joanne mentioned, as well as Jim, really trying to use that data to develop more manipulative or experimental kinds of understanding of the drivers that allows you to look at the drivers for occurrence and potential risk. And I guess going back to the model of food safety, right? If you've got a HACCP plan and you've got you understand your critical control points, when we get to that point, then that might be those critical control points and associated targets would be what we want to monitor very rapidly. But I'm not sure that that's the key gap right now. And I'm not trying to speak for everybody, but you guys kind of concur with that? I mean, because we're part of this group that's talking about detection and systems biology, right? So what we're saying now is that we've got a lot of ability to detect already, and we really need to translate that into understanding of the dynamics in the food production system so that we really can develop risk models. And more rapid analysis right now isn't the key roadblock.
I agree, Jay.
And I don't, Manon?
Yeah, I think you make a really good point. Like, it's not necessarily like identifying that critical control point using your HACCP example, but I think it's taking different types of data and trying to integrate them. Like we do a lot of work for enteric pathogen survival in the environment, in water and in soil. We can say, for example, okay, E. coli survival in soil that is amended with animal manure is dependent on a couple of their survival, their longitudinal survival can be dependent, say, like on the rainfall or the soil moisture or the type of amendment that you're putting down on the soil. But integrating that, say, with like, what's the weather pattern during that season? Like, okay, how much did it rain? What was the temperature? What was the specific source of manure? I think integrating all these different data sets gets us closer to that. And I think while it may be easy, I won't say easy is not the right term, but it's more straightforward, I think, to collect a certain type of data. It's maybe more straightforward to collect data related to water, right? Integrating that with the data related to soil, data related to the environment, and putting together those sort of I don't know if multi-dimensional model is the right term, but all those things that go into that Danielle was mentioning that can be interrogated by a machine learning thing, I think that's the challenge. And you have all these different targets that we're talking about. Like we spend a lot of time trying to target enterohemorrhagic E. coli and salmonella, but are we going to start targeting, say, levels of specific antibiotic resistant genes? And we get better at finding them and get more refined at doing that. Like, so I think like having to put it like in statistical terms, what's our response variable going to be? And how many different facets of environmental production, I don't know, seasonal weather, how many different things can we put into a model that doesn't just make our heads explode and we get that identifiable driver out of, I think, is our challenge.
So do you already have all those different data types that you were talking about? Like, have you already collected, weather patterns and all the different data types you were discussing?
So, I mean, we have some data sets where we look at survival of E. coli in soils with manure. And we have, the biological data. We have like the rainfall patterns. We have the daily temperature. I won't say that we're really good at, we're good at collecting sort of basic weather data. I don't know that we're good at, say, like wind speed and stuff like that. We don't have, but we have some of the basics, and we've put together, some good models with our statistical consulting service, the statistics groups here in ARS in the Northeast area. They've made some really nice models. We've tried to look at drivers of survival using like different levels of soil nutrients in E. coli and evaluate their transfer to cucumbers. So things like that we've been able to do. It's just how many different seasons can you evaluate like that? And we're limited by that. So, but yeah, we've done it a little bit. We have some publications on it.
Yeah, and I think the metadata that is going to be collected for any of these environmental samples, that's one of the areas of our work groups was on developing the metadata so that you could, like Manan said, effectively interrogate those samples then using the metadata as well as the response data with machine learning or other kind of multidimensional kind of analyses. And that's consistent with what we've been saying, I think. So I think there's general concurrence on that. On this idea that, yeah, we already can generate a lot. It's really got to move beyond the observational, get into this mechanistic understanding, and really translate that into these models, both decision support and risk models. Before we talk about those kind of models a little bit, since this is systems kind of focus, is there any area that is that we any kind of a field or tool that you think we have not leveraged enough of yet to really address this system level issue? Or do we have all the right kinds of scientists? We certainly have a diverse group here, right? Represented by you all, but are we missing anybody or any kind of talent?
I think we just have to get everybody together. right? And we need the folks who are in the AI world, and we need the folks who are in the methodology development world and the risk management world and our world. And one of the things that I wanted to point out, what we really need as well is sort of some standardized data collection and repository. If you all are doing that with your water samples, and we probably need to have a lot of conversations about what sorts of data. Like you said, we can collect a lot of data, but let's allow our data to talk to each other and be used together. And if we don't have some standardized way of sort of communicating that data, sharing that data, and I guess what are we saying? ARG levels, relative abundance. List any of them. It has to mean something. It has to mean the same thing to you as it does to me, as it does, to everybody else, for us really to be able to work on this full system and solve the system, solve the questions that we have together.
I wholeheartedly agree with that. That's why I asked Dr. Sharma, do you have the data? Because if all that data is in the database, the barrier is not the technology. We can today take, thousands and thousands of features and do multimodal, machine learning. It's not a problem. To do that, technology is not a problem, but standardizing a field and across a lot of different scientists, that's a challenge.
But I think we're getting closer. I mean, we have this Ag Commons platform within ARS, which is, I think, a really good start because it provides some standardization to enter sort of the metadata of where you collect your study and some of the things Jay was mentioning. It doesn't format the data, the format that you have to use, but I think stuff like that can be refined. I don't know that at this point you're starting at the beginning, but those common sort of public access where researchers can post their data and also get the acknowledgement for posting it, getting a DOI associated with those data, I think is a really good start. I'm definitely not knowledgeable enough what the next steps are, but we've put our data on that platform. And we want people to go use it. So if we can integrate all those things into what Joanne said of getting the right teams of people together, those people should definitely be included in the conversations on how to make their platforms more accessible and more to facilitate that data transfer.
Just to add, Lisa Durso in the Q&A, she's made some comments. I mean, she's affiliated with ARS's long-term agro-ecosystem research network, or LTAR, and that is, bringing together a number of different people, and they're trying to incorporate AMR data into other long-term agronomic procedures. And she's involved in a manure shed project, an LTAR manure shed project that's incorporating AMR into soil, environmental, chemical, physical, and they all also are strong proponent of standardizing data formats and building databases for sharing. So I think the point Lisa's making is that there's an example of what we've been emphasizing is that standardized both methods, but also formats for the data to enable this kind of higher level analysis of the factors affecting the distribution and occurrence of the AMR, the drivers. That kind of thing is going on in ARS through LTAR, and I think she points at that it's a good point to make that Joanne mentioned, you need to bring people together. Well, sometimes the way you bring people together is on an integrated project, right? So you make them kind of work together within that the project. So thanks for sharing that, Lisa. And then there was another question real briefly. It was a question for Walt, and he's not here, unfortunately. And the question is about azole resistant. Is it increasing given the concern about a human fungal disease there with the Aspergillus fumigatus? And the question is it increasing in ag? And I think there are some indications that it is. I don't want to speak for Walt, but that's something I have talked to some people about that. I don't know if anybody else online has knowledge of that, but it is an increasing concern because azoles have been, the use of azoles has been rapidly increasing in certain row crops, not just specialty crops like orchards over the last several decades. And because of that increased use and potentially increased selective pressure, there's some concerns about what the spillover might be to human health. So I think we're at the time now where I guess according to the original structure, we're kind of beyond the discussion phase and kind of in final remarks and just general thoughts. And I think some of that I've already summarized, and I think we concur that we've talked more on the need for really synthesizing these large data sets that we can already collect. The data does, the analytical and technology allows us to collect a lot of data. Certainly there can be improvements, but maybe the biggest need is really understanding the interactions in the data and what the drivers are and the currents so that you can better inform these risk and decision support models. Now, we haven't talked a lot about, what the framework of those are. We talked a little bit about HACCP and just the general plan for identifying critical control points, but Manon pointed out that that's can be difficult when you have more a less controlled kind of production system. So with more and more inputs like the environmental inputs that he's trying to characterize. But I think that's one thing. And so we really have emphasized the need to develop data in a coordinated way where you're bringing together different disciplines and having standardized approaches for the data, both production and synthesis, so that you can really interrogate that data with these advanced analytical tools like machine learning that Danielle mentioned to really get at understanding what the key drivers are. That's I think what we've all said is we feel like that's the key gap right now in the next, say, decade or maybe 5 years, right? See how fast we go. Any other, just quickly around the room, any other final thoughts that any of the speakers would like to point out? Because if not final thoughts, I guess one other way to phrase that is, kind of next steps. What do we, we kind of mentioned that this need, and Lisa brought it up, this need to really develop these integrated projects where you're trying to really bring people around studying a given system to understand it, like the LTARS. It's a good example. And I think the work that Jim mentioned is another example, really trying to look at watershed-based analyses effectively. I know one of the other, in the Q&A, someone had raised a question about another group in PNNL that we talked to about them, to them. And then there's a question online is there is a risk for producers for liability of AMR disclosure? And that kind of builds in from the producer side. Yeah, and we certainly have run into this, right? From the NARM side, there's sensitivity about generating data on the watershed side, right, on the surface water side. And what are you trying to attribute my production facility to that occurrence? And I think what we've tried to emphasize at this point is that we're really doing this to understand what the risks are, so we can inform further subsequent downstream, not downstream, no pun intended, downstream kind of regulatory actions of any kind. But that's not where we're at right now. But certainly the questioner is correct. There's a potential liability there, and there's a sensitivity from the producer side. But anyway, that said, any thoughts on key next steps other than what I've kind of tried to summarize? What do you want ARS to do over the next 5 to 10 years to address this issue?
Well, if I had my wish, it would be, money to run, human clinical trials.
Right. Back to your point about really doing the kind of manipulative kind of experiments. Yeah. Yeah.
Exactly.
I would say do simultaneous like assessment and mitigation, because it takes us a long time. These are complex problems, but I even think a little bit of data can be informative for mitigation and solutions. So we should really think about all the different scales of agricultural producers we have, fruit and vegetable growers, animal producers, and come up with maybe cost-effective solutions that can be helpful to those people who are making the food, as we produce food and grow food in more and more diverse environments and not just field agriculture anymore. And we can get those sort of solutions or treatments into the people's hands to mitigate some of these things, some of the trends, even if we don't know what all the trends are going to be at this time.
And in that case, the mitigation is somewhat of the manipulation, right? I mean, it's experimental studies, right? It's a good point.
As long as those mitigation strategies that are offered have a good basis in science, that we maintain our sort of reputation is not really the word I'm looking for, but we don't lose credibility by suggesting other options that we're not sure will work, that we're just hypothesizing will work.
Right. So the observational studies lead to the hypotheses that can lead to the mitigation, the potential mitigations. I think like your work clearly shows that, Joanne, and even Danielle, which she mentioned, she's ready to do manipulative studies, but that's based on some clear findings she's already seen with the trends within the relationship between diet and AMR in the microbiome.
And I think moving forward, I would think we sort of need more of this. I know Jim and Manon, and I've not met Danielle before, but we all need to keep talking in this space, because we learn a lot from each other. And I think if we continue to do that, we will help ourselves and each other's research move forward in a functional way.
Yeah, let alone bringing in interagency efforts, right, John?
Well, we certainly need that as well, and we do that.
That's it's been a pleasure working with Jim and Manon.
Yeah. So Jay, actually, with the interactions we've had, looking at it, there's some expertise on the EPA side from Office of Research and such, particularly on some of the modeling and the probabilistic design. It would be maybe even useful to have some more communications within your fellow scientists to work with some of these things. They bring a different perspective and a different tool set than many of us who in ARS have been working with. So,
right. Well, and on the other side, EPA is the agency that regulates the application of antimicrobials in crop agriculture, right? Not animal ag, but crop agriculture. So our Office of Pesticides is tasked with regulating and understanding what the risk is, and they do that now with limited understanding of the quantitative impacts. So it's more of a qualitative risk assessment, and they're very interested, and we are engaged with them over the next couple of years to try to develop more of a framework for how do we approach the risk assessment, more of a quantitative risk assessment for use of those antimicrobials in crop ag. So that's a really good example where communication between USDA-ARS and EPA-ORD would be very productive. So we're almost to the top of the hour. I'm always sensitive to scheduling. I guess our new goal in EPA is to not schedule meetings to the very end of the hour, but to give people at least a few minutes in between meetings. So if there's no other final comments, I would just say that it's been enjoyable. It was impressive kind of understanding the diversity of researchers with an ARS looking at this problem. And I think that we've concurred based on our dialogue that there really is a great opportunity to leverage the data that we can develop right now. We can collect right now. We need to do it. Use the kind of more of the observational studies we've done to date. Continue to build good observational data sets, but use those to lead to more experimental or manipulative experiments to really look at key drivers for occurrence and risk of AMR. And I really appreciate you guys giving me the opportunity to speak with you today, and it was a pleasure.